Imagine for a moment a single medical treatment that could fix thousands of different genetic diseases, everything from cystic fibrosis to Batten disease. I know, it sounds like something straight out of science fiction, right? Well, a really groundbreaking gene editing approach is making this a very real possibility. Today, we're going to dive deep into a new technique that doesn't just fix one tiny genetic mistake, but potentially an entire class of them. It's a discovery that could totally reshape medicine as we know it.
So, here's how we're going to break it down. First, we'll get a handle on just how massive this problem is. Then, we'll do a quick review of the basic biology, how our cells actually build proteins. After that, we'll get into the really cool part, the ingenious new strategy for hacking that process. We'll look at the incredible results that suggest this could be a universal fix. And finally, we'll talk about the real-world hurdles and the huge ethical questions on the path from the lab to an actual patient.
All right, so let's jump right in and focus on the core problem we're trying to solve. And believe it or not, it can be an error as small as a single misplaced letter in our genetic code, but one with absolutely devastating consequences. And to give you an idea of just how big a deal this is, get this. About a quarter, 1/4 of all known genetic changes that cause disease fall into this one specific category. So we're not talking about some rare, obscure problem. This is a fundamental challenge in genetic medicine that affects millions of people all over the world. It really does all boil down to this. One wrong letter gets swapped in our DNA. And that single mistake creates a premature stop signal where there shouldn't be one. The results? It can lead to some of the most severe inherited disorders we know of. The technical name for this kind of error is a nonsense mutation. Now think of your DNA as a super detailed instruction manual for building a crucial protein. A nonsense mutation is like a typo that sticks a full stop right in the middle of a sentence. The cellular machinery just follows the instructions, hits that stop, and halts production, leaving you with a half-built, totally useless protein. Okay, so to really get how the solution works, we first need a quick refresher on the basic biology that these mutations are messing with, the cell's very own protein factory. This is pretty much the central dogma of biology in action. First, the code in our DNA is transcribed into a portable copy, a message, called messenger RNA, or mRNA. That message then travels out to the cell's protein factory, which is called the ribosome. And there, the ribosome reads the message, and another molecule, transfer RNA, is the one that brings in the right amino acid building blocks to assemble the final protein. So, the really crucial player in all of this is this molecule right here, transfer RNA, or tRNA. You can think of it as the master translator. It reads the 3-letter genetic words on the mRNA and makes sure the exact right amino acid is added to the chain. And it's this translator that scientists have figured out how to hack. And that brings us to the breakthrough. We're now going to look at the incredibly elegant solution that scientists have engineered to get around those faulty stop signs in the genetic code. I mean, how do you tell a cell to just ignore an instruction that's literally written into its own genetic code? Well, the answer, it turns out, isn't to try and erase the mistake, but to give the cell a brand new tool to simply read past it. The core idea is just brilliant in its simplicity. Instead of trying to fix the faulty gene itself, which is really hard, scientists are using these ultra-precise genome editing tools to permanently change one of the cell's own genes, the gene that makes the translator molecule, the tRNA. They're essentially reprogramming the translator. This new strategy is called PERT. It uses a very precise gene editing tool called prime editing. You can think of it like a search and replace function for DNA, to rewrite one of the cell's own tRNA genes. This creates what's known as a suppressor tRNA. And this special new translator is the key. It now recognizes that premature stop signal not as an order to halt, but as a signal to add an amino acid and just keep on building the protein. Now, the concept is really powerful, but the big question is, does it actually work? Let's take a look at the evidence and see why this approach has generated so much excitement. The initial results from lab-grown human cells are, frankly, incredible. In models for several really devastating genetic diseases, the PERT strategy worked. It successfully produced the full-length proteins that were completely missing before. Enzyme activity, which was basically at 0, was restored to levels that could be genuinely life-changing. And the editing process itself was highly efficient and permanent in most of the cells they treated. And this right here, this is the most powerful takeaway of all. This therapy is gene-agnostic. You know what that means?
It doesn't care which gene has the nonsense mutation. It only cares about the mutation itself. This gene-agnostic quality means that the very same suppressor tRNA could, in theory, be used to treat thousands of different disorders, as long as they're all caused by the same type of premature stop signal. And look, it's not just us saying this. This potential is being recognized by experts in the field. As Dr. Debojochi Chakraborty points out, this isn't just a small step forward. It's a proof of concept for a whole new class of therapy, one that could totally change the landscape for treating these rare genetic diseases. Of course, moving from a proof of concept in a petri dish to an actual treatment for patients is a huge leap. And that brings us to the very real-world challenges and the profound ethical questions that we absolutely have to confront. A really critical distinction we have to understand here is the difference between somatic and germline therapy. Somatic therapy, which is what the PERT approach is designed for, targets the body's non-reproductive cells. The changes only affect the person being treated. Germline therapy, on the other hand, would edit reproductive cells like sperm or eggs, making genetic changes that would be passed down to all future generations. And that is a line that carries immense ethical weight. Yeah, the path forward isn't exactly a straight line. There are huge questions. On the technical side, how do we safely and effectively get these editing tools to the right cells inside the human body? What are the long-term safety risks? Are there off-target effects we don't know about? And then, ethically, the questions get even trickier. How do we make sure these incredibly expensive therapies are accessible to everyone who needs them? Where do we draw the line between treating a disease and enhancing a person? And the biggest one of all, should we ever make changes that are heritable, permanently altering the human gene pool? The science we've talked about today offers this incredible, tantalizing vision of the future, a future where one single, elegant, universal fix could correct a fundamental flaw that's responsible for thousands of diseases. The potential is absolutely immense, but the journey to actually get there will require not just scientific brilliance, but also deep, profound societal wisdom.